I'm Grant Gordon at the University of Calgary in the Department of Physiology and Pharmacology in the Cummings School of Medicine and a member of the Hotchkiss Brain Institute. In our study, we were interested to further our understanding of how and under what circumstances astrocytes become active in the conscious brain. Astrocytes are critical glial support cells that not only perform a number of essential homeostatic roles, but also actively modulate brain activity. Astrocytes are key players in the regulation of synaptic strength and microcirculatory blood flow. They perform these roles primarily through changes in free intracellular calcium at their fine processes which interact with synapses and at their end feet which wrap all parenchymal blood vessels. Now, a single astrocyte is ideally positioned to sense both local synaptic activity and the blood supply. Yet despite astrocytes' position to integrate inputs from both of these sources, our understanding of the factors that govern an elevation in astrocyte free calcium is limited.
Tran, and I am a postdoctoral fellow at the Hotchkiss Brain Institute at the University of Calgary, and the first author of this work. When neurons become active, this initiates rapid vasodilation of penetrating arterioles in the neocortex. This causes a relatively fast increase in blood perfusion that helps restore the energy deficits associated with neuroactivity. Here, we perform in vivo two-photon imaging on active mice to examine the calcium activity patterns of neocortical astrocytes during sensory evoked blood flow increases. We speculated that examining astrocytes in awake animals would be important to be able to observe when and how astrocytes become excited, because the brain and the microcirculation can interact in a realistic manner. We tested the hypothesis that to become activated, astrocytes integrate inputs from local glutamate synapses, broader neural network activity that relate to the animal's behavioral state, as well as inputs from the blood vessels themselves.
We utilized our awake in vivo mouse model, where the animals were head fixed but allowed to freely ambulate on an air-supported ball. A cranial window was created over the barrel cortex. We measured astrocyte calcium dynamics and vascular activity in response to a brief air puff to the contralateral whiskers using two different astrocyte crelines and genetically encoded calcium indicators.
We observed fast arterial dilation that was followed by a delay in widespread astrocyte calcium transient in the arbor, somar, and end feet. Interestingly, we found that peak amplitude of end-foot calcium transients was significantly dependent on the behavioral state of the animal. In particular, movement of the animal potentiated the response.
We then tested the contribution of synaptic glutamate by topically administering ionotropic receptor antagonists to the surface of the neocortex. These treatments strongly inhibited calcium transients in the end foot to whisker stimulation.
Examining the role of the vasodilator, nitric oxide, we found that a selective nNOS blocker decreased sensory evoked vasodilation but did not affect end-foot calcium. In contrast, putative blockade of eNOS increased arterial basotone and surprisingly reduced the evoked end-foot calcium signal.
Given that end-foot calcium followed peak dilation and eNOS blockade reduced the end-foot calcium signal, we hypothesized that increased blood flow itself contributed to end-foot activation. To test this, we used chemogenetics to directly cause vasodilation. Stimulating vascular GQ dreads generated large vasodilation that was followed by a delayed elevation in end-foot calcium.
We also used optogenetics to test the same hypothesis using a vascular inhibitory opsin. We found that light-induced vasodilation was again followed by an increase in end-foot calcium.
We hope that our work will provide insights towards how these glial cells participate in blood flow control and synaptic plasticity, as well as how changes in the integrative response of astrocytes contributes to disease.